This is the only way I could be in the middle if I had like. We each have different because these there's no room. And Hannah puts you know if you backpack under my chair every time.
Do you see how I'm trying to be nice? See, that was so like because no one likes it.
I just gotta keep the numbers.
You remind me of Jake Paul when he's on. So that's I didn't either. I didn't either. That's not a good thing. We had three dogs. We walked up to beat us and we said, because analysis down in the oh, that's what we do. I really know that. Them saying, you're dumb. Why? I mean, I
usually say,
yeah, I'm gonna have to.
I know, wait.
I'm so fair enough. Not that there's one where I really just went there and he just sat in his chair. The first person I saw was
Oh, yeah.
and it was before we were friends.
Of course.
I'll get back down.
Oh, that's cool.
I thought so. If you do the one, what do you mean by that? Yeah, like I went through this one.
Hannah, I have to.
I went through today, so I'm gonna go first. That's our sound.
I know how to work Excel, doesn't mean I know how to do Excel. I am like,
Why do you have to do
trying really hard.
it's not a zero, it's just like. Yeah. We're trying to also got here. Yeah, I pretty much that. That doesn't even work. I didn't want to see people in this was I go, okay, and administration was really good. I pulled my own
Every day.
Yesterday there was ice.
Oh no, I was so I have you were asking. I love that. You didn't ask you that.
No, did you further back? We didn't have it.
Right down there.
I'm gonna say this again.
Probably not. Nothing's funny then. Look the other way. Right? Stand here. So we should come over. He reaches over. Hold on. Okay, if you're out there, everybody has a video. Oh, it's connected. Wait, is this the double bond? Yeah, try double bond. Already? I'm pretty sure. Oh, so that's what we have to plus. Sometimes I feel like you have more estrogen than testosterone.
What?
That's how I feel about these people.
So that's why that is.
This one. This is America.
Oh my God. You felt like you want to get rid of that.
No, especially the fact that, like, what is he saying? No, I know it's so.
All right, guys, good morning. Happy Friday. All right, what did you guys think of the exam on Wednesday?
I'll tell you once I get my grade back.
Really good.
I thought it was pretty fair.
Okay. Yeah, the average so far was, because I still have a couple of people that need to take it, but the average so far was a 72%, which is,
We should ever thought they got higher.
So I changed my mind and I don't feel good.
so that's the average. And actually that's a great average.
That makes me feel really bad. I'm gonna be honest. Me too.
We had one person score 100%.
Yeah, so whoever that person is, we're gonna talk after class.
So it is the first exam. Yeah, for these types of classes, I just want to kind of orient you guys. An average of a 72% is actually good. That's kind of what I aim for. If it was below, I would have curved it to about a 72%. So since somebody did score 100%, don't hate that person.
Oh, I already do.
That I won't be curving it, but the final grades at the end of the semester will be curved to make sure that, you know, we have a good even distribution. Like I said, it is the first exam. You guys are still getting adjusted to my types of exams, my types of questions. So I wouldn't sweat it too much. I will pass them back as soon as everybody has taken the exam, and I'll put the answer keys up on Blackboard. And so once all of that is up, you guys cannot contact me for 24 hours until you've gone through the actual exam key and gone through what you got wrong and figured out why you got it wrong on your own or with your friends in the class. So after you guys do that, if you still feel like there's an issue or you feel like you want to contest any of my questions, then you're welcome to email me or come to me and tell me. Does that make sense? Okay. All right. So today we are starting Unit 2. You guys ready to learn about viruses?
No, I'm kind of stressed out. I'm gonna be honest.
I'm stressed. Yeah, I'm scared.
I mean, I'm telling you, don't worry too much about it. It's the first exam.
Please, that's the PhD, okay?
Actually, I taught this class last year, and it was a similar exam. I did change some of it, but last year, I was looking through it this time around, and I was like, man, that was brutal. I really made it difficult last year. This year, I thought I made it a lot easier, but you guys are obviously have your own opinions, and that's okay. But like I said, in a class like this, a 72% is not necessarily a C-. In a class like this, it actually equates more to a B. So I would keep that in mind when you get your scores back. And yeah, we'll go from there. Yeah.
Huh? Oh, I just had a question.
Yeah.
On, like,
What do you mean, like, wait, like, at the end of, like, the semester, do you do, like, that?
Yes, I will curve it. I will curve it so that people with, for instance, a 70% will end up getting, say, a B minus or a B. Yeah, so that's kind of how it works. Yeah. As soon as everybody takes the exam, I still have 2 more people that need to take it. One person is going to take it today. Another one is still sick, so when they take it, probably next week, and then I'll pass them back. I will put the scores up before then, so you at least can see your score. Unless that's going to make you guys freak out even more, then I don't want to do that. But I can put the scores up, but I can't give you your exams back to actually look at what you got wrong until everybody takes it. Okay. All right. Any other questions, concerns, comments? All right. So today, guys, I'm gonna need your attention today. Please, Ben, need your attention today, please. All right. So does anybody recognize what this photo is up here? The thing in the blue, what's the blue? There's 2 parts to this picture. There's the blue and there's the orange.
Virus and the orange the phage.
Close. Yeah.
The bacterial cell and the orange is a phage.
Yep. So the blue is a bacterial cell, the orange is a virus or a phage. Phage, which I'll tell you about, are basically viruses that can infect bacteria. So bacterial cells are actually normally much larger than viruses, and so that's why you see this bacteria being infected by all of these phage. So today we're gonna dive into viruses, some readings in chapter 6. We'll talk about what a virus is, specifically viruses that infect bacteria. So these bacteriophage, also just called phage. We'll talk about the genomes and how they're structured. And so on the left, I'm showing you corona. If you guys didn't know, coronavirus is named coronavirus because corona actually means these spiked like proteins on it. So that's why it's called coronavirus. If it has these like infamous spiked, these proteins, then it's a corona type of virus. And so you have, on the right, you have a more common looking bacteriophage that kind of looks like what you guys saw here. We have like the capsid head and then this sheath and then these tails that actually attach to the surface of the bacteria and allow for the injection of the DNA, the viral DNA into the bacteria. We'll then talk about the 3 life cycles of bacteriophage, the lytic cycle, lysogenic cycle, and the slow release cycles. So just some nomenclature for you guys. A virus, it's considered a microbe, but it's not alive. So it's basically not a living cell, so it's a non-cellular particle, but it has to infect a host cell in order to reproduce and survive. So it can't reproduce autonomously or independently, and so that's why we don't consider a virus living. If it can't use its own cellular machinery to replicate and reproduce, then it's not a living thing on its own. And so, well, what it can do is exploit the cellular machinery of that bacteria or of its host in order to replicate itself. There are viruses that infect specific cell types, and all cells can have some type of virus that can infect them. But there are specific viruses that have specific host ranges. So there are some viruses that can only infect bacteria, some viruses that can only infect humans, but all cell types can be infected by a virus. A virus particle is called a viron. You'll hear that a lot. So if I'm asking about a viron, I'm asking about one virus particle. And every virus particle has some sort of nucleic acid, and that is usually protected within a protein capsid. So the nucleic acid can be DNA or RNA for viruses. And every virus will have to have that DNA or RNA to be encompassed in some sort of capsid, which is usually protein. It's a protective outer layer. Does that make sense? Yeah.
Is the protein called capsid or is, okay.
Yeah. So the protein is usually called the capsid. It's just the cap, essentially. So all viruses will have nucleic acid and a capsid. Some viruses have more features, but not all of them do. Before we start going into depth into the bacterial viruses, there are some taxonomic features that we should talk about. And so in green, we have these eukaryotic viruses that have DNA as their genome. So some of them can be double-stranded, some of them can be single-stranded, but there are all of these different viruses have DNA as their genome. Remember, it could be DNA or RNA. So herpes virus, papilloma virus, adenovirus, all of those. You don't have to remember any of those names. The only one you'll have to remember is coronavirus, which I'll talk about here at the end. But there are some that have DNA as their genetic material. And that genetic material can be either positive sense or negative sense. Also, that is something you guys don't need to understand in depth, but just for your own curiosity, if a virus has positive sense DNA or RNA, it means that the viral RNA is the same sense as the mRNA. So remember, in transcription and then translation, you have mRNA, and then when you translate it into protein, you basically have to have your messenger RNA going from the 5' end to the 3' end. So it's just saying that if it's positive sense, when that viral RNA gets integrated into that host, it can immediately be translated by the host cell machinery. If it's negative sense, that means the RNA is complementary to the RNA sequence, so at first has to be converted to the positive sense, and then it can be translated with the cell's protein machinery. You don't need to remember all of that except for the coronavirus. So in orange, those are the viruses that have RNA as their genetic material. So once again, it could be single-stranded or double-stranded RNA. And then here we have some negative sense ones and some positive sense ones. So the positive and negative sense usually is associated with the RNA, not the DNA. So as you can see, all of these viruses right here, they'll have DNA and then they have a protein capsid. It can be filamentous, it can be just circular, it can be corona-like, where you have these spikes. So this right here is coronaviride, so the coronavirus. This one is single-stranded RNA in the positive sense. Some viruses can be enveloped or have a membrane, or they could be naked. So enveloped or naked are the two terms we use. So if it's enveloped, It's gonna have an envelope around the protein capsid, but if you have just the protein capsid, then it's non-enveloped. You still have that protein capsid, you just don't have that envelope on top. Capsids can range in shape. Like I said, they can be filamentous, they can be icosahedral, they can be circular, they can be even more sophisticated than that. And some of the capsids can assemble in the nucleus or in the cytoplasm. You don't need to know too much about the assembly site in the host, but viruses can either, make their genetic material and become a virus and inject either in the nucleus or in the cytoplasm. But once again, the one you should know is the coronavirus, and so here's a concept check with the answer right on the slide. Coronaviruses have which of the following features? Although I didn't tell you if it's enveloped or not, but you should still be able to, yeah.
A.
Yeah, A. Coronavirus is enveloped. It has single-stranded RNA, and it's in the positive sense. Make sure you write down A, because I'm not going to have the answer on here. Yeah.
So all viruses have that capsid. What is the envelope? How is it?
Yeah, that's just another added layer.
So would that be the spikes that surround it?
No, the spikes are not necessarily the envelope in this particular one. It's not shown here in this picture. It's just some other protein or like sugars and things that are just around the protein capsid. Yeah. Any other questions? Let's let that sink in for a little bit. Okay, so now to the fun part. Enough about those eukaryotic viruses. Now we're going to talk about viruses that can infect bacteria. Bacteriophage is a virus that infects bacteria, also known as a phage. Basically, they are bacterial eaters, as the name suggests. The way that we study bacteriophage in the lab is by putting the bacteria on a plate. So here in the yellowish color, that's all bacteria on the plate. And then you put the phage on top, and so essentially you'll see the bacterial cells being eaten away and broken down by the phage, and so these empty circles are actually where the phage was able to infect the bacteria and kill the bacteria. So we talked about bacteria growing as colonies, and so it's kind of the inverse of this. You actually see like a colony of cells on the media. This is the opposite, where you have bacterial cell death, and that's how we study phage in the lab, by using plating. So this is called a plaque, and this is where you have the lysed cells on a lawn of bacteria. So when we have the bacteria not as colonies and it's just all over the entire plate, we call that a lawn. And another thing you guys should know is that in the very center of each one of these plaques, you can assume that there was one single phage that infected the bacteria in each plaque. So each one of these, there's one phage particle, one viron that infected the bacteria surrounding it, ate away at it, and formed a plaque. Many of phage are tailed. So they essentially have these tails at the end. So this is a bacterial cell, this is a phage. And so their genomes, they vary. The DNA or the RNA can vary. Like I said, they can be single-stranded or double-stranded. So this is just reiterating the same thing as before. Before we were talking about eukaryotic viruses, viruses that can infect like the human host. These are viruses that can affect bacteria. So they can also be single-stranded, double-stranded, DNA or RNA. Sometimes it can be circular or linear. And they're typically smaller than the actual cells. Any questions so far? Yeah.
So there's plaques. There are obviously multiple different bacterial cells. Is there any real reason, obviously it could be done by just one, but does it have to be done by just one viral factor, or could multiple happen at the same time?
Yeah, that's a good question. So generally the assumption is that if you see a plaque, it took at least one. So that's why we say one,
right.
Because at least one. In theory, yeah, it could have been more than one particle, but it takes just one to form a plaque. Right. So you see there's some overlap here. So when you see things like this, then that's probably multiple. So that's something you could say for certain.
Right.
You can't say for certain because the way you're analyzing this type of result is by looking at just the plaque. The plaque is going to be circular. It's going to end at some point. So the virus ate away as much as it could, and then it stopped, so you make a plaque. So that's kind of why we say one particle.
Okay.
But good question. I had that question before, too. Any other questions? Okay, so just to give you some context, to put things into perspective for you, and just to tell you about how abundant phage are on Earth in comparison to other living things, the number of human cells that we make is about 37 trillion. We have about 37 trillion human cells, and we actually have about the same number of bacteria in us and on us. Remember from Unit 1, we are just as much microbial as we are human. We have so much bacteria on us. The number of bacteria on Earth is just ridiculously higher. So if you were to count all of the bacteria on Earth plus every living thing on Earth, that's not even close to the number of phage on Earth. So the number of phage on Earth is about 10 to the power of 31. So that's 1 with 31 zeros in population. That's just the different types of phage on Earth. They're incredibly diverse and abundant. There are 1 billion more phage than stars in the universe, just some more perspective. 10 billion more phage than grains of sand on Earth. And 250 million phage in 1 drop of seawater. So with so many phage on Earth, should we be worried? Let's talk about that a little bit. Should we?
Probably not.
Probably not. Yeah.
There's not, because we're not all constantly sick, even though they're everywhere.
Yeah. Yeah, so we actually shouldn't, because phage only have mechanisms to infect bacteria. Remember, phage is a virus that infects bacteria. It's a bacteriophage. So there's no direct harm to human cells, eukaryotes and human cells, but they infect bacteria. And if I just told you that we are actually half microbial, we have a lot of bacteria in us and on us, then of course they have indirect effects to human health. So they can affect us indirectly. That's something that we can't miss. But like I said, they only have mechanisms to infect bacteria, so they have some sort of specificity. So over here, you can see that the specificity actually comes from these tail fibers. So now we're going to start talking about the actual structure of the phage and why that they're important. But the specificity comes from basically these tail fibers recognizing certain receptors on cells. So these are just color-coded. If these are 3 different cells, one's red, one's yellow, one's blue, there are phage that will have red, essentially, quote-unquote, tail fibers that will recognize the red, yellow will recognize the yellow, blue recognize the blue. They're not actually colors in real life, but just to give you a little bit of an idea of how that works, there are receptors that are exclusive to the surface of certain bacterial cells. So the receptor is on the phage. It's going to receive information when it connects to the bacteria. Phage perform bacterial genocide every single day, essentially. So they're very host specific. A specific phage can only really infect certain types or species of bacteria or those that are very closely related to it. Any questions? Okay. So phage structure basically has some roles. First, it's to keep the genome intact, that DNA or RNA, and it also helps to enable the infection of the right host cell. So like I said, the phage require 2 things. They need to have some sort of DNA or RNA, some sort of nucleic acid, and it's going to have a protein capsid. So that satisfies number 1, to keep the genome intact with the protein capsid. And then enabling the infection of the right host cell is really important in that specificity of the tail fibers. The capsid packages and protects the phage from the genome from degradation. But phage can have very different forms. So they don't all have these tails. They can have, they don't have to have tails and they still have that specificity. The bacteriophage we'll talk about mostly will have those, spider legs, those tails, but they can either be tailed, they can be tailless, which we just call icosahedral because you just had that icosahedral protein capsid without the tail. Or they could be filamentous, so they could look like filaments, like we talked about some bacteria looking like. You don't need to know the specific names, but if I were to show you a picture of one, you should be able to tell me if it's tailed, icosahedral, or filamentous.
So the tails from the viruses, obviously they help kind of latch on to the bacterial host. How do the filamentous and the icosahedral actually grab onto their?
They have other attachment proteins and receptors on their surfaces. It's just not in the form of the tail, but there are other mechanisms that they have to be able to attach to the surface. Yeah. So this is a major category of viruses, the filamentous viruses, the most famous actually being Ebola. And you can see the DNA in green in there, and it actually has this long tube around it of protein, and so that's the protein capsid. So here, there's this protein on the end of this. So to answer your question, Ben, there are certain proteins that attach. These are actually pili, similar to bacteria, and that will allow it to enter the host cell. So they have those proteins on both ends of it. Filamentous viruses will vary in length, and that depends on the DNA, because it's actually it's supercoiled along the entire length of what that protein capsid can hold. This is an M13 phage. It's just one of the ones that's more widely studied in the lab setting, but you don't need to know too many of the details about it. But yeah, so just that's how you have that attachment. And so for filamentous viruses, the length of the genome dictates the length of the virus. At the end of these, you have those tail fibers, those protein fibers that will mediate the specific attachment to the bacterial cell.
For these, is there like a specific like DNA or RNA that these are, or can it be any?
It can be either, yeah. It's just this one is M13, this one is DNA, single-stranded, so that's the example here, but it can be both.
Okay.
All right. So tailed phage, they have an icosahedral capsid head, and they actually have this really elaborate delivery device, which consists of a tailed sheath. That's the thing that's used as a delivery device to deliver the DNA or RNA, the genome, through and into the cell. The tail fibers are used for stability, stabilize the phage. So this is a really cool image of E. coli being infected by this phage. So we're going to talk about T4 phage specifically. T4 phage have this icosahedral head with a really supercoiled genome pressurized in the head, this tail sheath, and these tail fibers. So I want you guys to familiarize yourself with the structure and some of these names. The head contains the pressure-packed DNA or RNA. For T4 bacteriophages, it's double-stranded DNA. Then you have this neck, this sheath, that will eventually channel or deliver the genome into the bacterial cell. Then you have the tail fibers that stabilize the phage onto the host cell surface. So over here we have an E. coli cell, and you can see the heads, you can see the sheath. You can't necessarily see in this image due to low resolution. You can't see the actual tail fibers that are attached. But it's pretty cool to see using a certain electro image, EM image. Okay, so concept check. What do all viruses contain? Yeah. C, a capsid and a genome. Does that make sense to everyone? Okay. So we actually know mostly about bacteriophage that are found in the gut. Obviously, if there's an association with human health, then we're going to want to know more about the biology of those phage. So gut bacteriophages are often called coliphages. So coli, like we talked about in lab, or coli, like E. coli, stands for colon. So that should tell you a coliphage is a gut bacteriophage. And so this is important. It's more important to study coliphages because they, can, as a result, modulate our health indirectly. And so they can have implications in our digestion, in our immunity, in our mental health. So research out there actually shows that phage may be able to limit the bacterial numbers to levels that our immune system can tolerate. So they can also have helpful implications for us. So they can indirectly help us as well. They control the diversity of microbes in our gut. Phage can also attack, destroy, and dissolve biofilms. You guys remember what a biofilm is? What's a biofilm? Yeah, I heard that. Basically, they are communities of bacteria that are attached to each other and to surfaces, so they can actually attack biofilms. Okay, so this is some background on just phage, their structure, their genomes, some things that you need to know. And so we're next going to start talking about how a phage can start an infection of a bacterial cell. So there's going to be contact and attachment, and we'll talk about the receptors that are specific. Then we'll talk about the actual genome then being pulled out of the capsid head and through that sheath, and so it can be delivered into the bacterial cell. And then it uses the bacterial cell's machinery once it's in there to replicate its genome and make more protein for the capsid head, and then it'll proliferate in the bacterial cell, eventually killing it. Like I said, there's 3 life cycles: the lytic, lysogenic, and slow release cycles. But I'm thinking I'll probably stop here, let you guys digest all of this, because we just abruptly switched over to viruses. And so next time we'll start talking about how phage can start an infection and fulfill it.